Hi, I'm Carol Leibel, and today we're going to examine how prokaryotic cells regulate genes in the expression of their proteins. This is a biochemical pathway in which a precursor molecule undergoes a series of enzymatic reactions to produce a product. That product is the amino acid tryptophan. And as you can see, there are 5 enzymes that are needed in order for tryptophan to be made. It would be entirely wasteful if a cell had lots of tryptophan to transcribe these genes and to continue making tryptophan. It would be most efficient if a prokaryotic cell had a way to activate and deactivate these genes. Well, it turns out that they do. Now, there are 2 different ways that we could regulate this biochemical pathway. One method is to have some sort of molecule either activate the gene or deactivate the genes. The second method is if this product or one of these products along this biochemical pathway could interact with that enzyme or another enzyme and deactivate that enzyme. If the enzyme is deactivated, then it's not going to be functional and the biochemical pathway is going to be halted. You may be asking yourself, how does eukaryotic gene regulation compare with prokaryotic gene regulation? Well, take into consideration that eukaryotic cells have many more genes than a prokaryotic cell. For example, humans have 23,000 genes compared to only 3,000 genes found in a prokaryotic cell like E. coli, a bacteria. In a typical human cell, there's over 6 feet of DNA packed into a cell that can't be seen with the naked eye. Now, in order to manage 6 feet of DNA in that very small space, eukaryotic cells actually wraps the DNA around proteins called histones. Prokaryotic cells do not have that much DNA. So it's not necessary for them to take their DNA and to wrap it around histone proteins. And so they don't have to worry about managing all those histones. In addition, there are other non-histone proteins that are found in eukaryotes that are not found in prokaryotes. Now, I've got to say that prokaryotes are a lot more efficient in how they regulate their genes. Remember, we had an example in which we were looking at the production of the amino acid tryptophan, and we said that there were 5 enzymes involved in that. What is pretty unique and cool about prokaryotes is that those 5 genes that are needed to make the 5 enzymes are physically right next to each other. This is not unusual in prokaryotes, and when you have a group of genes sitting side by side that function in the same biochemical pathway, that group of genes, they're called an operon. Now, what's cool about an operon is that in order to activate these genes, there has to be one on switch, if you will, and that on switch is called the promoter region. It turns out that an operon only needs one promoter region. So in this example here, this one promoter region is going to activate all 5 of these genes. Now, a promoter region is where the RNA polymerase is going to attach and start the process of transcription. So an operon is composed of the following. It's composed of structural genes that are related and used in the biochemical pathway. It includes a promoter, and a promoter are the nucleotide sequences that can attach to the RNA polymerase to start the process of transcription. Within the promoter region, there's an operator region, and an operator region is a nucleotide sequence that can attach to something called a repressor protein. Now, what a repressor protein can do is it can attach to the operator site and it can physically prevent the RNA polymerase from starting the process of transcription. You might be asking, how in the world are repressor proteins made? Well, a little distance from the operon is a regulator gene. A regulator gene produces a protein called a repressor. And as I mentioned, oops, let's go back. As I mentioned, a repressor protein has the ability to attach to the operator, and it prevents the RNA polymerase from transcribing the operon. Now, what is cool about repressors is that they have allosteric properties. Do you remember from the beginning of the year what if a protein has allosteric properties, what that means? Take a minute and think about it. If a protein has allosteric properties, that indicates that that protein has the ability to take different conformations or different shapes. Now, in order for that protein to change shapes, what has to happen is that a molecule is going to attach to it. Those molecules that will attach to the repressor protein to change its shape is called a modulator. So here are two different scenarios of as to how that's going to work. First off, here is a repressor protein. And in this case, in this particular state, it's active. However, when this molecule attaches to it, it's going to change shapes, and now it's going to become an inactive repressor, and it won't work. Now, here's another way that it could work. This here is the repressor protein. However, this time when the molecule attaches to it, it becomes active. So in this state here, without the modulator, it's inactive, and when it attaches, it becomes an active repressor. Now, because that molecule, when it attaches to it, makes it an active repressor, that modulator has another name. It's called a co-repressor. So we're going to look at an example of an operon system that involves the repressor. And the first one we're going to look at is called the lac operon. Now, remember that lactose is a disaccharide carbohydrate, and that carbohydrates can provide energy to the cell. But in order for a bacteria to use lactose as an energy source, it has to digest the lactose. It turns out that when you ingest lactose, you have to do the very same thing. But in order to break down lactose, it's going to require a biochemical pathway. And this biochemical pathway has three enzymes. The first enzyme is beta-galactosidase. This is the enzyme that actually breaks lactose down. And by the way, when lactose is decomposed, it's decomposed into glucose and galactose. And here is another enzyme that's involved, and that enzyme is permease. The permease affects the permeability of the membrane so that it allows the disaccharide to enter the cell. And the last enzyme is transacetylase, which, to be quite honest, they're not quite sure what that does. As we mentioned, repressors are often involved. So what is going to happen with this repressor if lactose is around? Allolactose, which is an isomer of lactose, will attach to the repressor protein. And what that does is going to make the repressor protein inactive. So if lactose is around, lactose inactivates the repressor protein. RNA polymerase has the ability to attach to the operon, and now transcription is going to take place. Messenger RNA is going to be made, and the proteins are going to be produced. Now, if lactose is around, the operon is going to be transcribed. Basically, lactose, when you think of it, turns on the operon, or it induces the operon to be active, if you will. And so therefore, the lac operon is said to be an inducible operon. Here's a little animation. You've got your regulatory gene. the repressor protein is going to be made. It's in its active form. Allolactose, an isomer of lactose, attaches to the repressor protein. It becomes inactive. This allows the RNA polymerase to attach to the operon. The genes are going to be transcribed and translated to make the enzymes necessary in order to utilize lactose as an energy source. All right, so let's look at what happens if lactose is not around. If lactose is not around, this repressor protein stays active. If it stays active, it's going to attach to the operator region. Remember, when it attaches to the operator, it physically prevents the RNA polymerase from transcribing the operon. So here's a little animation showing that happening. The repressor protein is made. No lactose is present. It's going to attach to the operator region. RNA polymerase is prevented from transcribing the operon. So now we want to look at this lac operon, and I'm sorry, we're going to have to go out onto the internet in order to examine this, so please have some patience here.
Transcriptionally regulated. Such regulation is a critical feature of both eukaryotic and prokaryotic organisms. One of the classical systems used to investigate transcriptional regulation was originally investigated by Jacob, Monod, and Pardee. Their studies involved the transcriptional regulation of the genes used to ferment lactose in the bacterium Escherichia coli. 3 genes clustered together on the chromosome are required for the bacterium to utilize lactose. These were determined to be the LAC-Z gene that codes for the enzyme beta-galactosidase, the LAC-Y gene that codes for a lactose permease, and the LAC-A gene that codes for the enzyme galactoside transacetylase. The LAC-P region is the promoter that is needed to transcribe the LAC-Z, LAC-Y, and LAC-A genes as a single polycistronic mRNA. The LAC-O region is an operator site that is involved in the transcriptional regulation of the LAC operon. The unit consisting of the LAC promoter, LAC operator, and the LAC-Z, LAC-Y, and LAC-A genes is called the LAC operon. Located near, but not in tandem with, the LAC region is the LAC-I regulatory gene that codes for an mRNA that is translated to produce a protein referred to as the LAC repressor. In the absence of lactose inside the cell, the LAC repressor protein is active. In its active state, the LAC repressor recognizes and binds to the LAC operator site. When the LAC repressor is bound to the LAC operator site, RNA polymerase is prohibited from recognizing the LAC promoter, and the LAC-Z, LAC-Y, and LAC-A genes are not transcribed. When lactose enters the cell, a small amount of it is converted to allolactose via beta-galactosidase. Allolactose binds to the LAC repressor. This causes a conformational change in the LAC repressor protein that prevents it from binding to the LAC operator site. Inactive LAC repressor is unable to bind to the LAC operator site. Without an active LAC repressor bound to the LAC operator site, RNA polymerase is able to bind to the promoter, and a polycistronic messenger RNA is transcribed that includes the LAC-Z, LAC-Y, and LAC-A genes. This polycistronic messenger RNA is then translated to produce the beta-galactosidase, lactose permease, and galactoside transacetylase proteins. Therefore, the induction of the LAC operon enables the bacterium to efficiently transport lactose into the cell and metabolize it.
All right, so I hope that helped with your understanding of the lac operon. The next thing we want to look at is another example of an operon system, but it's called the Tryp operon. And we've already mentioned the Tryp operon at the beginning of this presentation. It's the Tryp Operon is involved in the synthesis of the amino acid tryptophan. Okay, and so as you can see, 5 genes are involved, okay, and 5 enzymes are needed. Now, the repressor protein with no tryptophan present is in an inactive form. And now what's going to happen is with no tryptophan, it cannot attach to the operator site. The RNA polymerase is going to attach, transcription is going to take place, and all 5 enzymes are going to be synthesized. Here is an animation showing you this process. Okay, the repressor protein is inactive, but there's no tryptophan around. So the RNA polymerase is going to attach, transcription is going to take place, all 5 genes are going to be transcribed and translated into the enzymes necessary to make the amino acid tryptophan. All right, but let's suppose tryptophan is around. If tryptophan is around, it can attach, let's go back. It can attach to the repressor, and when it attaches to the repressor, it makes the repressor active. When the repressor is active, it can attach to the operator site, and it will prevent the RNA polymerase from attaching to the promoter region, and it will prevent transcription from taking place. Here is a little animation showing you that process. Tryptophan's around. Remember, it's going to act as a co-repressor. It attaches to the repressor. It becomes active. Now it's going to attach to the operator region. It's going to prevent the RNA polymerase from attaching and transcribing the 5 genes. And so it stops the synthesis of tryptophan. All right, so we have a little animation that will hopefully clarify it a little bit further.
In the bacterium Escherichia coli, a group of 5 genes code for enzymes required to synthesize the amino acid tryptophan. All 5 genes are transcribed together as a unit called an operon. An operon is a group of genes that is under the control of a single operator site. A regulatory protein called a repressor can bind to the operator site and prevent transcription. When tryptophan is lacking in the environment, the repressor is inactive. RNA polymerase binds to the promoter site and then proceeds down the DNA, transcribing the genes for the tryptophan biosynthesis enzymes. When tryptophan is present in the environment, the organism no longer needs to make tryptophan. Tryptophan binds to the repressor and activates it. The activated repressor now binds to the operator located within the tryptophan promoter and blocks transcription. The tryptophan repressor is a helix-turn-helix regulatory protein. When tryptophan is absent from the environment, the repressor is in an inactive conformation and cannot bind to the DNA to prevent transcription. When tryptophan is abundant, 2 molecules of tryptophan bind to the repressor. This alters the orientation of the helix-turn-helix motifs in the repressor and causes their recognition helices to fit into adjacent major grooves of the DNA. Thus, the synthesis of tryptophan occurs when it is needed, but is repressed when tryptophan is available.
All right, thank you for your patience, and I hope that helped further your understanding about the Tryp Operon. The one thing I did want to remind you is that because when tryptophan is present, the transcription of the operon is stopped or halted, the Tryp Operon is called a a repressible operon. All right. Now, both lac and Tryp operons have repressor proteins, and repressor proteins will halt the transcription of the operon. So because a repressor protein halts the transcription of an operon, that's said to be an example of negative gene regulation. As I mentioned before, eukaryotic genes have, in general, they are involved in positive gene regulation. However, I did want to show you an example of some positive gene regulation. Okay, and this involves the lac operon again. All right, sorry that this is so messy, but this is the way it works. First off, there's another gene involved in the lac operon, and it produces a protein, and that protein is called the cyclic AMP. It's called the cyclic AMP receptor protein, or the CRP protein. Now, the CRP protein also has two different forms. It can be active or inactive. Now, it turns out that let's go back. Sorry, back again. It turns out that when lactose is present and glucose is scarce, that causes an abundance of cyclic AMP to be around. Now, cyclic AMP can attach to this CRP protein. And when it attaches to the CRP protein becomes active. When the CRP protein becomes active, it attaches to a region in the promoter region. Now, instead of being inhibitory, what happens is it's kind of like this sign that says, "Hey, RNA polymerase, over here," and attracts the RNA polymerase. And so now the RNA polymerase will be attracted to that region, and now transcription will take place. Okay? Now, let's suppose lactose is present and glucose is present. Okay? So glucose is present. Now, if glucose is present and you had a choice, which should I eat or which should I use for energy? Lactose, the disaccharide, or glucose, the monosaccharide? Well, for most cells, the choice is simple. Glucose is easier to use as a source for energy. Now, when glucose is around, remember that cyclic AMP? It turns out that it's not around. So if cyclic AMP is not around, what happens is that this becomes inactive, and so that's not there to help the RNA polymerase to attach. All right, again, because this protein, the CRP protein, when it attaches, helps the RNA polymerase attach and helps transcription take place, this is said to be a positive control for gene regulation. All right, so that's the end of our little discussion about prokaryotes and gene regulation. I hope that helped, and I hope I see you as we discuss eukaryotes and how they regulate their genes. We'll talk to you later. Take care. Bye.